Okay, Chapter 17, Fear, of Bitten, The Secret History of Lyme Disease and Biological Weapons by Kris Newby. Washington, D.C., 2001. Camp Dietrich was born of fear. It now helps to generate more fear and is thereby itself regenerated. Theodore Roseberry, Chief of Airborne Infection, Camp Dietrich. Your scientists were so preoccupied with whether or not they could, they didn't stop to think if they should. Jurassic Park. On February 13, 2001, William Patrick III, the former chief architect of Fort Detrick's biological weapons program and Joel McCleary's friend, gave a public talk to the threat of biological warfare at the Washington Roundtable on Science and Policy. On his business card, he described his current job as a biological warfare consultant. He was the ultimate insider on the threat of biological weapons, and he was there to offer his advice on how to protect the country from what he considered to be real and present danger. To be a real and present danger. He was essentially profiting from fear. Patrick started the talk by opening his briefcase and displaying its contents to the audience. The sample case I brought with me today holds glass bottles containing exact stimulants of the weaponized form of anthrax and virus causing Venezuelan equine encephalitis. Now I've carried this case through a number of airports on my way to meetings over the last 12 years, but no one ever stopped me and asked, what are these peculiar-looking powders? He went on to describe military vulnerability tests conducted to educate our leaders about the threat of biological weapons. He showed the audience a small plastic spray bottle. He said it was capable of spraying large particles of a liquid biological agent that would be relatively easy for terrorists to produce. Yet, when I traveled through airports, no one has ever stopped me, Patrick said. To further prove his point, he spoke about the big metal handheld spray duster that he packed in his suitcase. It could disseminate very small particles, 1 to 3 microns in diameter, small enough to evade the human respiratory tract's defense mechanisms. The particles could lodge inside the tiny sacs within the lungs, causing a deep-seated infection. I have been through all the major airports in the country. I have been through X-rays at the CIA, DIA, Defense Intelligence Agency, the State Department, and the House of Representatives, and nobody ever stopped me to ask what I'm carrying. The guards at the security points do a good job of stopping people with knives or pistols, but they don't have a clue as to what a biological weapons terrorists might be carrying around, Patrick said. Then Patrick described some large area coverage vulnerability tests that the military conducted on an unsuspecting public over the years. In the 1950s, Operation Sea Spray, U.S. Navy boats sprayed a 2-mile-long line of aerosolized stimulant off the coast of San Francisco. He described how effective such an attack could be if the weather conditions were right. Patrick neglected, however, to tell the audience that this supposedly harmless stimulant, Bacillus subtilis, a spore-forming bacterium, had sent 11 people to Stanford Hospital during the trial because of serious urinary tract infections. One of these patients, Edward J. Nevin, an elderly man with a compromised immune system, died 3 weeks later. He described another stimulant attack on the Elgin Air Force Base located on the Florida Panhandle. An open-air hog cholera virus test conducted in July 1951. Patrick, our mock attack was expected, and guards and guard dogs were posted around the perimeter, but we did not have to go to the base. We attacked from a road about a mile outside the security fence, behind the thicket of shrub pines between us and the base. If you've been down to the Carolinas of Georgia, you know how thick these pine trees grow. yet the aerosol passed through them without any diminution, went through the housing area and contaminated the hangars and the airplane cockpits. Samplers showed high levels of contamination, and finally Patrick described a naval test off the coast of Alaska, explaining why an aerosolized attack can be done in stealth mode, leaving very little evidence behind. We sprayed BG spores, Bacillus globigi, another live bacterium stimulant, about 20 miles upwind of ships with samplers. When the aerosol reached the ships, it was pulled in by the air system and remained at very high concentrations for about an hour and a half. We found especially heavy concentrations of spores in the engine room, which pulls in very large amounts of air to dissipate heat, but the exterior and interior surfaces of the ships were only very lightly contaminated. The same air system that brought the aerosol in removed the organism with very little residue having fallen out. There were similar findings in other tests where a building was contaminated with a primary aerosol. It comes in at high concentrations. Start this over. There were similar findings in other tests where a building was contaminated with a primary aerosol. It comes in at a high concentration, gets you infected, and then departs, leaving very low residue. People become infected because the human body is nothing more than a vacuum pulling in that aerosol. A little more than 7 months later, after Patrick's talk, the 9/11 terrorist attack happened, followed by the deadly anthrax mailings. Allegedly perpetrated by Bruce Evans, a microbiologist and biodefense researcher who worked at Fort Detrick. This raises the provocative question, was the timing of the anthrax mailing a pure coincidence, or did Patrick's fear-inducing talk motivate the mailer, whomever it was, to demonstrate how vulnerable the country was to such an attack? Outbreak. Chapter 18, The Fog of War. New York, 1966. A single automobile spraying germs across the country through its tailpipe could cause an epidemic that could destroy crops, kill off livestock, or wipe out hundreds of thousands of people. Jack Anderson, Montana Standard Post, August 30th, 1965. On June 6, 1966, an invisible man who looked like everyone and no one stepped into a crowded subway car at the 14th Street station in Manhattan. He was of average height and build. His thinning hair was combed over his cue ball head. He wore a cheap suit and dark sunglasses. What looked like a photographic light meter hung off his belt, and he carried a plastic briefcase that emitted a faint whirring sound. The man was Charles Sensini, a CIA weapons developer from the Special Operations Division at Fort Detrick, and the leader of a 21-person team running a covert operation to see how vulnerable New Yorkers might be to a bioweapon attack. As he rode the subway car, one of his operatives stood at street level over a subway ventilation grate and opened the brown paper bag he was carrying. As an unapproaching train rumbled beneath his feet, he pulled out a light bulb and shattered it over the grate. Upon shattering, it released an invisible, odorless cloud of bacteria that was sucked into the tunnel by the passing train and rapidly disseminated through the whole network of tunnels. The cloud held approximately 87 trillion spores of Bacillus subtilis, a bacterium thought to be harmless. It had been freeze-dried and processed into particles that mimicked the physical properties of weaponized anthrax. For the next few hours, Cincinnati's team rode around the subway carrying bacterial sniffers disguised as briefcases and purses. Cincinnati's photographic light meter was actually a device that tracked temperature and humidity. At the end of the day, one of the sensors at the 23rd Street station showed calculated respiratory exposure to be 100,000 spores a breath 5 minutes after the light bulb broke. By June 10th, a million New Yorkers were hatching spores in wet warmth. By June 10th, a million New Yorkers were hatching spores in the wet warmth of their lungs, said Cincinnati. Had it been anthrax in the light bulbs, the spores would have put New York out of commission. This was one of the many open-air tests conducted in the 1960s and 1970s. The CIA and the U.S. Army and the Department of Defense. The coastal tests were conducted by personnel in Project Shipboard Hazard and Defense, SHAD, SHAD, who sprayed simulated a live biological and chemical warfare agents over the North Atlantic and Pacific Oceans near the Marshall Islands, Hawaii, Puerto Rico, and the California coast. Land-based tests took place domestically in Alaska, Hawaii, Maryland, and Florida, Utah, and Georgia, and internationally in Panama, Canada, and the United Kingdom. In 1964 and 1965, they used Bacillus subtilis to simulate the physical characteristics of the smallpox virus in airborne tests in Washington, D.C.'s National Airport and Greyhound bus terminal. Some of these human experiments were revealed through the Senate's 1976 Church Committee report and an independent Church of Scientology investigation and a 2003 class action lawsuit filed against the U.S. government on behalf of test subjects and veterans involved in SHAD projects. But a few of these open-air tests are still classified, the records have been destroyed, or the details of the operations were never put in writing. In its 1966 budget report, the Pentagon noted that 57 U.S. universities and affiliated research institutions were among the top 500 defense research contractors. At the same time, the anti-Vietnam War movement was being fomented on campuses, and students and scientists were beginning to question their institutions' involvement in chemical and biological weapons research. One of the most vocal critics was Joshua Lederberg, Ph.D., a 1958 Nobel Prize recipient for his pioneering work on bacterial genetics while at the University of Wisconsin. After he moved to Stanford, Lederberg began early research on gene splicing and started to understand the responsibilities that can come with creating new life forms. The concern motivated him to start lobbying policymakers to draft a treaty to ban biological weapons. The large-scale deployment of infectious agents is a potential threat against the whole species. Mutant forms of viruses could well develop that would spread over the Earth's population for a new Black Death, said Lederberg in a Washington Post editorial on September 24, 1966. He added, "The future of the species is very much bound up with the control of these weapons. Their use must be regulated by the most thoughtful reconsideration of United States and world policy." A month later, the Army's Biological Subcommittee Munitions Advisory Group thumbed its nose at this national pronouncement made by prominent scientists, downplaying the scientists' concerns. Fort Detrick's scientific director, Riley Housewright, said that such publicity would probably best be considered to be an annoyance. The advisory group then continued discussing its plans for genetic manipulation of microbes, new rickettsial, and viral agents, and the development of a balanced program for both incapacitating and lethal agents. Even that rickettsia was being tested as a possible lethal agent was hiding in plain sight in lab safety studies published by university researchers. In 1966, one of the experiments was contracted out to Samuel Salslaw, a professor of infectious disease at Ohio State University. For this military-funded study, Salslaw mixed 68 monkeys with aerosolized droplets of a lethal dose of Rickettsia rickettsii, a disease that the study's authors noted is spread in nature only by tick bites. They described the symptoms of the first monkey to die. On the fourth day of exposure, its temperature rose to 104.2 degrees Fahrenheit. On the sixth day, the temperature spiked to 105.8 degrees Fahrenheit. On the seventh, the monkey sat quietly in its cage with its head hanging in its arms, clasped around its body. On the eighth, it coiled with the large, it coiled, it collied with the side of it collied with the side of its cage and fell down. Its heart sounds were weak and indistinct. On the ninth day, it experienced a delirium, extreme weakness, and dehydration. On the 10th, it went into a coma and died. One photo showed a necrotic monkey's paw, human-like, with blackened nails and fingertips. At the end of the article, the researchers concluded Rocky Mountain spotted fever was produced in 56 of 60 rhesus monkeys following exposure to aerosols. 42 of 56 died. Since the disease so closely simulates that observed in naturally occurring infection in man, one should be aware of this potential route of infection, partially among laboratory personnel. On March 13, 1968, during a chemical weapons experiment, a spray nozzle on a military aircraft malfunctioned over Dugway Proving Ground, allowing VX nerve gas to kill a flock of sheep in the Skull Valley area. It could have been worse, especially if that cloud had drifted over the heavily populated Salt Lake Valley. The incident was an international scandal. The reputational damage to the chemical and biological weapons program could not be undone, and the accident set in motion a chain of events that would end in President Richard M. Nixon terminating the U.S. offensive biological weapons program on November 25, 1969. More than 2,000 sheep died in 1968. Nerve invasion occurred near Dugway Proving Ground in Utah. Nixon sold us down the river, said Murray Hamlet, DVM, former director of research at the United States Army Research Institute of Environmental Medicine. Hamlet was working with Fort Detrick when most of the bioweapons researchers were let go with no warning. We had the best immunologists in the world, and all of a sudden they had to find new jobs. What were they supposed to do? Start selling ice cream in town, he asked. As the projects under the bioweapons program began winding down, Willie was optimistic that he could restart his research on spirochetes, but his NIH director refused, saying no one cared about a disease that could easily be cured with antibiotics. As a consolation prize, Willie began investigating rickettsial outbreaks in South Carolina, Tennessee, and Kentucky. He was also assigned to the Armed Forces Epidemiological Board's Commission on Virus and Rickettsial Disease. This commission, run out of Walter Reed Army Institute of Research, had previously worked on the offensive bioweapon program. But once the Nixon decision took effect, it shifted emphasis to defensive projects, vaccines, protective measures, and detection systems. But the little loophole that they have, we need the deadly aerosolized pathogens to know how to fight against them. During this transition, Dale Bergdorfer had a lot of ups and downs. She would tire easily and retreat into silence. Because of his concern for her, Willie had to cancel a trip to a Rickettsia symposium at the Czechoslovakia Academy of Sciences. Then on July 21st, 1969, Willie's mother wrote, "My dear Willie, 8 days ago they picked up your paw around 2:30 from our house. Forever." His father had leaned a ladder up against a cherry tree in the pouring rain to yell and swipe at a flock of birds trying to eat his ripe cherries. The ladder slipped. He fell and hit his head on a stone. Later he suffered a fatal heart attack. Now the birds have their cherries, Willie's mother wrote. Chapter 19. Lone Star, Newport News, Virginia, 1968. There's no such thing as a clean tick, Willie Bergdorfer. On August 28, 1968, Daniel E. Sonnenschein, Ph.D., 35 years old, slight of build with an impish smile, walked up to a swampy, wooded area of Newport News, Virginia. Gnats, mosquitoes, and biting flies swarmed. The air throbbed with the grating sound of fluorescent green katydids rubbing their wings together. It was late summer in the coastal tidewater region of Virginia, a place that felt like a steaming sauna full of bugs. Sonnenschein was an associate professor of parasitology at Old Dominion College, was launching another study on behavior of different species of ticks that carry Rickettsia rickettsii. In 1966, he'd studied the American dog tick, Dermacentor variabilis. This year he was studying the Lone Star tick, Amblyoma americanum, so named because of the shiny white star-like dot on the backs of females. Over 30 years, Sonnenschein had raised hundreds of thousands of ticks and released them in Montpelier and Newport News, Virginia, and in 2 canyons near Hamilton, Montana. With help of the Atomic Energy Commission, he also refined a technique for tracking the feeding and migration patterns of ticks in the wild. He made the ticks radioactive. In a letter, Willie taught Sonnenschein how to breed a large number of ticks. You put a rabbit in a linen bag with the adult ticks, and then, after 24 hours, the time necessary for the majority of the ticks to attach, transfer the rabbit to a wire cage and slide a large linen bag over the cage to prevent the engorge of ticks from escaping. After Sonnenschein's ticks had fed and mated on a rabbit, he carefully picked off the females that were about to lay eggs and pressed them into a strip of clay laid across a microscope platform. Looking through the microscope lens, he used a 13-gauge syringe to inject the body cavity of each pregnant tick with a sugar solution spiked with radioactive carbon-14 or iodine-125 liquid. The females would go on to lay a clutch of 1,000 to 3,000 eggs, and each larval hatching would be radioactive for the remainder of its 3-stage life. For the Newport News study, he planted poles of For the Newport News study, he planted poles to partition the woods into 47 equilateral squares, placing live animal traps covered with sticky tape at evenly spaced locations. 1,000 Lone Star larvae were then released inside each square. Over the next few months, Sonnenschein and his helpers would return to the woods to collect ticks from captured animals, cloth flags dragged around the ground, and the sticky tape. Each harvested tick was placed in a vial labeled with the location of the square in which it had been captured. Back in the lab, a technician would place the vials under a scintillation detector to measure how many original release radioactive larval ticks were in the batch. Adult and nymph stage ticks were marked with colored enamel paint and then released into the square where they had been captured. The paint would allow them to be tracked as they migrated. Over the next 3 years, 194,150 radioisotope tagged Lone Star tick larvae were released at the 2 Virginia sites. Uncontrolled tick release, 1966-1969. These sites were located on the Atlantic Flyway, the migratory bird superhighway that runs along the eastern South America and North American coasts. On the face of it, there were clear public health benefits to these tick field tests. The Lone Star tick had been moving northward in the last few years, and it would be useful for the pest control people to know the rate at which the species was migrating. But the studies were also so useful to the U.S. military planners at Fort Detrick, who wanted to know how far Lone Star ticks might spread when released into enemy territory. Before the experiment began, there were no formal environmental impact studies done and no hoops to be jumped through. Beyond attaining a city permit in 2018, Sonnenschein admitted that it would be extremely difficult to get such a field test approved today. The Lone Star tick is a vicious biter, attacking man readily and voraciously, said Glenn Coles, K-O-H-L-S, Coles, the tick zookeeper who worked with Willie when he first arrived in Montana. The Rocky Mountain lab occasionally sent batches of Lone Star ticks to Fort Detrick. The immature stages are particularly annoying, since the intense itching produced by their bite may persist for 2 to 3 weeks. The adult, with its long, piercing mouth parts, is capable of producing a severe and sometimes painful lesion, which may require several months to heal. Yikes. Lone Star ticks have several survival advantages over their deer tick cousins. They don't wait patiently on a stalk of grass for passing prey. They are active hunters and crawl towards any carbon dioxide emitting animal, including birds. They swarm, and unlike deer ticks, they have primitive eyes that help them creep towards prospective prey. All 3 life stages of the Lone Star tick feed on foraging deer, which can carry them several miles a day to new wooded areas. God, reading about this just makes me fucking itchy. Jesus. Another tick expert from Old Dominion University, Holly Gaff, PhD, recently conducted experiments on Lone Star ticks and was surprised at their resilience. One batch survived 7 days in a 0 degrees Celsius freezer. Other batches lived after being submerged in fresh water for 70 days and brackish water for 64 days. Lone Star ticks carry several serious human diseases, including Rocky Mountain spotted fever, tularemia, and tularemia. Lone Star ticks carry several human diseases, including Rocky Mountain spotted fever, tularemia, heartland virus disease, and 2 species of Ehrlichia, Ehrlichia, a close bacterial relative of the rickettsia. And more recently, some people bitten by Lone Star ticks have suffered from a delayed reaction, long-lasting meat allergy caused by immune system hypersensitivity to the ephagal sugar molecule found in Lone Star tick saliva. Let's read that again. And more recently, some people bitten by Lone Star ticks have suffered from a delayed reaction, long-lasting meat allergy caused by immune system hypersensitivity to the aphagal sugar molecule found in Lone Star tick saliva. Even more worrisome, Lone Star ticks are on the move, replacing long-standing native tick populations. After World War II, Lone Stars were fairly concentrated in a region south of Mason-Dixon line, bounded on the west by Texas and on the east by the Atlantic coast. But in the 1970s, these ticks began rapidly expanding their range. The first Lone Star tick observed in Montauk, Long Island, what a story for there, was in 1971, and as of 2018, established populations have been observed as far north as Maine. All this begs the question, what is driving this mass migration of Lone Star ticks and its disease-causing hitchhikers northward? Climate change? A rise in deer population? The movement of humans into tick habitats? The release of ticks into new areas by scientists? Radiation-induced tick mutations? Or some combination of all these? No one knows for sure. In short, it's complicated. Chapter 20. Springfield, Virginia, 1969. If mankind were to disappear, the world would regenerate back to the rich state of equilibrium that existed 10,000 years ago. If insects were to vanish, the environment would collapse into chaos. E.O. Wilson, American biologist, theorist, and naturalist. When I was in 4th grade, my family moved from California to a rural area of Northern Virginia, close to my father's military work in Washington, D.C. This was the 7th place I'd lived in years. I don't remember making any friends that year. I was so thin, almost invisible, that boys in my class called me Toothpick. The creek where I spent most of my free time on weekends, I would pack up my frog hunting gear, a coffee and a fishing net made from a coat hanger, and my mother's nylons, and run across the countryside with my white blonde hair floating in the air like an untethered strands of spider silk. I'd cross neighbors' yards, which were separated by split rail fences, cut across tract home developments sprouting up from the rust-red soil, and finally arrived at the path that led to a nearby farm. Most of the farm was wooded, covered in the canopy of trees so dense that it was always dark beneath it. The air under the canopy was heavy and smelled of damp tobacco. I walked along the creek. I lifted the mossy rocks, looking for crawfish. I'd sneak up on brown salamanders with red fern gills sprouting out of the sides of their heads. Occasionally I'd find a black and yellow polka-dotted newt hiding in the damp leaf litter, a box turtle with orange eyes that practically glowed in the dark. After a half a mile or so, the creek fanned out into a series of ponds or sunny grass fields. This was my hunting ground. I'd start out trying to catch bullfrogs. At a distance, I'd echolocate them through their territorial croaks, and then sneak up and lunge for them with my bare hands. I was rarely fast enough to catch one. My consolation prizes were tadpoles. I'd scoop them up with the net and bring them home to my aquarium to watch them sprout legs and arms as they turned into frogs. One day, on the way home from the creek, I spotted a praying mantis egg case attached to a twig. It was a beige frothy orb about an inch in diameter, with the density of styrofoam. I snapped off the twig and put it inside my aquarium with the screen top and waited. A month or so later, I was leaving with my family on a week-long vacation. Several hundred baby mantises began emerging from the bottom of the egg case, forming a writhing rope to the bottom of the aquarium. I threw in some mealworms and left. When I returned, there was only one praying mantis left. Pieces of its siblings' arms, legs, and heads were strewn across the bottom of the aquarium cage. The sole survivor, its gullet bulging, saw me and began swaying back and forth, challenging me like a boxer, waiting to throw a punch. It cocked its head, and its green, soulless eyes looked up at me. I let the mantis free in my backyard. I wanted it out of the house, not staring at me as I slept. It was my first lesson in the Darwinian survival and the tenacity of insects and arachnids. I think back on those outings. I don't remember ever being bitten by a tick, but it was 1969. Lone Star ticks hadn't arrived in Northern Virginia yet. That was about to change. Let's see if I should continue. Chapter 21, Castleman's Case, Boston, Massachusetts, 1973. Since the 1970s, a series of unanticipated outbreaks of microbial disease startled inhabitants of the United States. Richard M. Krause, former director of the National Institute of Allergy and Infectious Diseases. In 1973, Benjamin Castleman, cheery and apple-cheeked with a few wisps of hair floating above his mostly bald head, queued up the first slide in the weekly clinico-pathological conference held in a classroom at Massachusetts General, the teaching hospital of Harvard University. It was a photo from the neck down of a prepubescent girl wearing a white cotton bra and underwear. An 11-year-old girl was admitted to the hospital with a 104-degree fever and a rash, said Castleman. This was a Castleman case, a stump the expert extravaganza for Harvard physicians in training. Later, a summary would be published in the New England Journal of Medicine. Castleman continued with the girl's story. The patient was well until 6 days before admission after a camping trip on the north coast of Boston with other Girl Scouts. Ticks were found on several of her companions, all of whom remained well. None were seen on a patient. He said that over the next 15 months, anorexia occurred with a daily rise of temperature as high as 103 degrees Fahrenheit, and a pinprick rash all over the body, muscle tenderness, and a weight loss of 11 pounds. 3 days before admission, the girl complained of aches in the knees and elbows and a shaking chill. 2 days before hospital admission, her temperature rose to 104 degrees Fahrenheit, and there were small, flat red spots over her ankles and soles of her feet. She complained of a mild frontal headache, and the evening before she was admitted, she vomited. Tests for rheumatoid factors and lupus were negative. Then she ceded the floor to Dr. Richard Masters, a young assistant dermatologist who was the discussant in the host seat of the session. Masters nervously moved to the front and looked out over a room full of medical professionals in white coats, observed from the walls by framed portraits of distinguished Harvard clinicians. He told them that the blood tests showed anemia, a mildly low white blood count, and low chloride. This was counter to what was typically seen in a severe bacterial infection. Masters cleared his throat and continued, I come back to the exposure to ticks. Although Rocky Mountain spotted fever is indigenous to the Cape Cod area, including Martha's Vineyard and Nantucket, the frequency of the disease has increased over the years. He added that the spotted fever is spread by ticks and occasionally by airborne particles. So what is your diagnosis, Dr. Masters? Said Castleman. Masters cleared his throat and said, spotted fever and juvenile rheumatoid arthritis in remission. Next, Dr. Stanley Rawboy, a young pathologist, took the floor in Castleman's world. Next, Dr. Stanley Rawboy, a young pathologist, took the floor. In Castleman's world, pathologists, the seekers of truth who find wisdom through the lenses of a microscope, had the final word. A microscopic image of a biopsied rash pustule from the girl's foot filled the screen. Raboy showed several slides of cross-sectioned blood vessels in the skin and then stated the microscopical examination disclosed a marked lymphocytic infiltrate that cuffed almost every blood vessel in the papillary and reticular dermis. A large artery in the subcutaneous dermis was occulted by an organizing thrombosis. And occulted just means hidden. Come on, people. Where was I? More simply, the cells that lined the girl's blood vessels were massively infected with rickettsial bacteria. Damaged vessels began leaking blood into various parts of her body, the skin, the lungs, and the brain. As blood began clotting in the fine capillaries of her brain, she experienced headache, a stiff neck, and sensitivity to light. Without treatment, she could experience convulsions, followed by a coma and perhaps even death. Raboy agreed. It was spotted fever, despite the fact that the antibody blood tests and cultures of blood, cerebrospinal fluid, and skin were all negative for the disease. The patient was started on intravenous administered tetracycline therapy shortly after the biopsy. 2 days later, she was discharged, Raboy said. Another physician chimed in about an unusual aspect of the case. At the time of admission, there was considerable discussion about the fact that Rocky Mountain spotted fever had only been reported in South Boston, in the vicinity of Cape Cod Islands. This was the first warning flare sent up by one of the most respected medical centers in the nation about the unusual outbreak of Rocky Mountain spotted fever. This version of spotted fever didn't show up on any standard tests, so there were probably many undocumented cases. A year and a half later, the CDC issued a limited distribution. A year and a half later, the CDC issued a limited distribution memo about a worrisome spike in Rocky Mountain spotted fever cases on the southern east end of Long Island. In 1974, there were 20 cases compared to only 5 in 1972. Then in 1976, Suffolk County on Long Island set a new record for spotted fever. 40 confirmed cases and 4 deaths. In April alone, the island's state health department received more than 3,000 complaints of heavy tick populations. There was also an unusual infestation of larval deer ticks on a nearby Martha's Vineyard, 3 months earlier than ever recorded. Peter Colt Joseph, a historian and author on Martha's Vineyard, sent an angry letter to the National Institute of Health complaining about non-responsiveness of the National Institute of Health, the Center of Disease Control, Harvard, and the Massachusetts Department of Public Health to whatever he viewed as a dire health crisis. Willie was copied on this letter. And we'll pick this up tomorrow or some other day when I feel like it.